Hello, folks. Thanks for joining us. My name is Erin Abram. I'm an associate college lecturer at Cleveland State University in the Department of Chemistry, and I'm here today with Dr. Kimberly Jackson. She is chair of the Department of Chemistry and Biochemistry, professor of biochemistry, and the director of food studies at Spelman College in Atlanta, Georgia.
Thank you. Thank you for having me.
Dr. Jackson is the primary author on a research article titled Dibenzoylmethane Induces Cell Cycle Deregulation in Human Prostate Cancer Cells, published in the journal Cancer Letters. Today, we'd like to chat about your hypotheses from that research study and some of the data published in this paper. We're going to start with the data shown in Figure 2A, which focuses on the growth inhibitory effects of DBM. Can you orient us to this graph?
At the time I was doing this work, I was focused on anti-cancer therapeutics. And so there's a basic algorithm to drug discovery. First, we have to figure out our model that we're going to use and what type of cellular model. And in that particular laboratory I was working in, we were using human prostate cancer cell lines. And then when you decide or you have your particular drug of interest, you want to do what we call proliferation studies. And so this was our first really big experiment to see whether or not this particular drug that we were working with caused any type of growth arrest. And this is an MTT toxicity experiment. Would you like me to unpack it a little bit further?
Yeah, so what were the key findings from this data, from this graph?
So from this particular graph, we found out that DBM at certain concentrations caused growth arrest or it impacted cell viability, which was huge. So we saw growth inhibition in this particular graph.
Excellent. Now, there are lots of different types of model systems. How did you choose this particular model?
I was actually working in a lab with someone who was a medical doctor and worked on prostate cancer patients. And so we decided to choose LNCAP because it was what we considered a traditional prostate cancer cell line because it had the androgen receptor and it was from a patient who had metastatic prostate cancer. And so that's why we chose this one. And we purchased it from the American Tissue Type Collection out of Rockville, Maryland. So that's where a lot of these cell lines are stored. And so we purchased that particular model.
Excellent. So let's move on to the next figure. So this is the flow cytometry data for DBM in this LNCAP cell line. Now, can you tell us about the data and what's being shown here?
Yeah, so again, like I said earlier, there is an algorithm for doing drug discovery work. So we had already done the cell proliferation data that you saw previously. Now it's time to move on and look at cell cycle analysis. So this is a flow cytometric data. It is a histogram. And flow cytometry is just a technique that we use to study cells. And the wonderful thing about doing flow cytometry, you're able to characterize cells, you're able to look at cell size, you're able to look at shape, granularity, and the presence of certain proteins or other markers. Here we're looking at DNA content. So if you think about the cell cycle, you have G1S, G2M. The cells have gone through because we've labeled them with some type of dye. We see scattering effects, about 10,000 units of cells. And you see this large curve in the G1 of the cell cycle. And so that's an accumulations of cells in G1. And then you have a small one in S and then G2. So you see at our control, we have our control with no dibenzoylmethane. And then in the second graph, there is 50 micromolar of dibenzoylmethane. And what we see there with the 50 micromolar of dibenzoylmethane is a slight deregulation of the cell cycle. And that's what you have.
Excellent. So taking this data in conjunction with the MTT results, what types of conclusions were you drawing here?
So at this point, we were thinking that, dibenzomethane actually was able to cause some type of growth arrest, stop the cell cycle at a particular point in the cell cycle. And so now we knew that because we saw this type of growth arrest, we saw this proliferation to go down, that we need to do some further studies around this particular drug and prostate cancer.
So what type of work did you do or do you think would be really useful to build on these results?
So some more work that students could do is to particular protein or gene. And we know that the androgen receptor is important in terms of the progression of prostate cancer. So now you can see whether or not the androgen receptor, that protein, is being impacted. So the next step for me would be maybe to do a Western blot or analyze that particular key protein or gene. After you start analyzing key proteins and genes, you may want to move into mouse model work. So this experiment can go further and further and further. It won't stop. This was just the beginning.
Excellent. And one of the interesting things about DBM is that it is a natural product, right?
It's a natural product. It's actually a minor constituent of licorice root. And our laboratory was the first to actually document the quantitative analysis of that and confirm that it was actually from licorice root.
Oh, excellent. Yeah, that is really interesting. So do you think that this will be a viable anti-cancer agent for prostate cancer?
I do, because as we move further along with this project, we did do some mouse model work, and we saw that it did stop the growth of these tramp mice that were supposed to get prostate cancer, and they did not. So we think it is a viable um, anti-cancer reagent.
Excellent. Yeah, that's really fascinating. So now I'd like to ask you a few questions about being a scientist and how you became a scientist. So what kinds of scientific questions interest you the most?
Yeah, so I love questions around proteins. How do proteins fold? What happens when they fold on themselves? And the DNA dogma that you learn in biochemistry. So that has always been so big to me. So even in my graduate studies, I worked with proteins of the immune system. So I've always been a big protein person. And now I started working on a particular protein, which was the androgen receptor as a transcription factor. So, yeah, so I've always loved proteins and why they work. So, yeah.
So when and how did you know you wanted to be a scientist?
Oh, wow. Yeah. I started college thinking I was going to be a flutist with the New York Philharmonic. So I
Mhm.
was a music major. And I went to my first week of classes in the music department. And boy, was it difficult. It was hard. They were asking me what were the different chords. Well, I had no idea. So I ran to the science building, and because chemistry was actually pretty easy. And I had the best time. At that time, people were talking about they wanted to be either a physician or a biomedical engineer, which both topics sounded pretty cool. So that summer of 1989, I went to what they call a health careers opportunity program at the University of Oklahoma. And there you shadow doctors. So this was a program for students who thought they were going to medical school. And I realized very quickly that was not for me. In my cohort, people were fainting when we saw babies being delivered. It was just a lot to stomach. And so for me, I knew that maybe instead of working with people, I can work behind the scenes to figure out how and why do these people have diseases, and maybe I can kind of fix on that. And so it was that summer that it came to me that I wanted to be a researcher.
Excellent. Now, do you still play the flute?
I do.
Did you
And
continue playing in college also?
I did. I played with what they call the symphonic band, and I had private lessons. And then I would play with the local symphony, like guests appear. But yeah, and throughout I played for weddings, and now my eldest daughter plays, and so sometimes we play together. And so yeah.
Oh, that's so special. I was actually a music minor in college also.
Oh, wow. Wow. I think we use both sides of the same side of the brain for science. So
Yeah, yeah, fantastic. All right, tell us about some of the paths that led you to your current job.
yeah, so when I left graduate school, I went on to get my postdoc, and I was doing my postdoctoral fellowship at Emory, and this is where I did some of this work. And I thought I wanted to be a policy person. So I went to the Centers for Disease Control for 1 year, where we would sit in meetings and make decisions impacting people's health. But I really wanted to not just sit in an office. I wanted to talk about this work and wanted to teach this work. And I'd always said I would never be a professor because my parents were educators. And I always told them that they were poor and I never wanted to be poor. And so I said that growing up. And then after that first year doing a lot of policy work and a lot of writing and meetings, and we were meeting about people of color and some of the changes I felt that the policies that were changing or being made were not going to be impactful or helpful to people of color. So I made the decision I wanted to teach and I wanted to have my small lab. And so I ran into teaching. And so I started in 2002 as a professor at Spelman.
Excellent. And you've been there. You've been there most of
I've been there ever since. I have moved through the ranks. I started out as what they call kind of like a visiting professor or teacher postdoc before a teacher postdoc was even a thing. And I was in that position and I moved into a tenure track position and I was promoted. But a part of my work, I was also a Fulbright scholar. I did that and I taught in Antigua and I did research there. And I was also a faculty affiliate at Harvard University School of Medicine, where I had a laboratory up at Harvard for some summers. And so, yeah,
fantastic. Those must have been amazing experiences.
wonderful experiences. My children got to experience it with me and my husband. And so, yeah, so it's been great. It's been
Yeah.
great.
Can you share a moment when you felt like you really belonged in the field of science?
Yeah, so there were several times I felt like I didn't belong. And there were even times where I went to this museum exhibit and I saw this exhibit by Carrie Mae Weems. She's a Black woman photographer that takes these beautiful pictures. She has a series called Museum Series, where she's standing in this long black funeral dress outside of these different museums like the Tate or the Guggenheim, some of these large museums. And she's standing there because she's feeling as though her works are not welcome in that space. And many times, as a Black woman scientist, when I would go to these large meetings, I would feel the same way as well because it wasn't very many of us. But it wasn't until I saw Carrie Mae Weems' museum series that it resonated with me. And this may have been in 2013, so not so long ago. This resonated with me, and I realized that not only did I belong, that it was my job to make sure other Black women or women of color feel like they belong as well. So there are certain words that I don't use and I don't believe in, like imposter syndrome. Um, I think sometimes what people are feeling is not imposter syndrome. Maybe they're feeling structural racism or microaggressions or biases. And I think a lot of times we use words to make the person feel as though it's their burden, and it's not. And so, because of now feeling as though I belong, There are words that I don't use because I do believe language matters.
Yeah, fantastic. That is such an important message. Now, what keeps you motivated to continue your research?
Oh, wow. So it is exciting. I have two arms to my research. I have the DBM prostate cancer arm, but I also have the arm of advancing equity for Black women scholars. And I teach at a Black women's college. So, um, it's exciting to share the work around centering spaces for Black women, whether they're scientists or whether they are food scholars, and making a space for them to feel as though they belong. So I love that part of my research, but I also love going into the lab and dusting off my stuff and say, okay, let's try to make sure we figure out a cure for cancer. And the excitement that students feel, because in this day and time, everyone either has experienced or they know someone who's experienced, or there's 1 or 2 degrees separation of someone who's experienced or been impacted by cancer. And so I think that resonates with my students. And so it's easy just to come in and say, hey, we're going to solve cancer, And so we get excited and we figure out pathways and mechanisms with these natural products to figure out how to do that.
Now, what's a typical day like for you?
A typical day starts about 6 a.m. 6 a.m. at my home. I check in with sometimes my eldest daughter, who graduated from Spelman and went on to Johns Hopkins, but now she's a U.S. diplomat in another country. So we check in because she's 12 hours difference, and so I may check in with her. I have two other children, one who's a graduate student in infectious disease. So I'll check in with him. That's the son. And then my youngest, who's actually a senior in my department in chemistry at Spelman. And I may check in with her and see how are things. And this is not every day. This is just at least once a week. But every day I get up, I give gratitude for being here. And I give gratitude and um, for enjoying life in my job and having passion in what I do. I say good morning to the husband. And then my day gets to rocking and rolling about 8 a.m. with meetings, meeting with students, um, sending emails, writing proposals. I try to get in the lab at least once a week for a couple of hours. And so, yeah, it's very busy. It's a very, very busy. And that's the exciting part of it. It's never boring. There's always something new every day.
Now, what is it like being a scientist who identifies as a Black woman?
Yeah, I think I would say it's special, but I don't want it to be an anomaly. I want it to be something that when people see me, they just say, "Oh, yeah, that's a scientist." But I think it's so hard now because in most people's minds, they may picture someone else. And so I'm excited. I get excited about it. And for me, it's just the norm. And it should be like that for everybody.
Yep, absolutely. Now, how do you balance work with your other interests?
So everything kind of moves together. And I even tell young faculty as well, sometimes we want to compartmentalize everything that we do, and we will never have enough time to do everything. So sometimes we have to use our classroom to do our research. And so, take for instance my biochemistry laboratory class. That became a part of my research. I think it's kind of similar to what you're doing. And so that makes it easier um, when you are doing activities. Why not bring in your family to support and appreciate what you're doing? Sometimes when I go to conferences, especially when my children were younger, they would travel with me. And so being able to integrate work and personal life has been wonderful because I think it makes it easier.
Yeah, fantastic. Yeah, my daughter went to a conference with me this past summer. I was like, let's go, kiddo.
Let's go.
Now, what's the most important advice you would give to young scientists?
Yeah, advice. I would say be free to be free to create, be brave. A lot of times today, we don't take the time just to be present. So be present, be free to be, and be brave.
What an important message. Yeah, that's fantastic. All right. Thank you so much, Dr. Jackson, for chatting with us today. If you would like to learn more about this work, you can check out the publication Dibenzoylmethane Induces Cell Cycle Regulation in Human Prostate Cancer Cells. This was published in the journal Cancer Letters. The citation will be included below this video.